This podcast is about organizing intermediary metabolic pathways by asking 5 distinct questions about each of them. The first question is, what is the pathway for, or what is its cellular purpose? The second question is, where does the pathway occur, in what tissue does it occur, and in which cellular compartment? The third question is, what is the energy output or input for the pathway? The fourth question is, what are the rate-limiting enzymes for the pathway? And how are these rate-limiting enzymes regulated allosterically and hormonally? And the fifth question is, are there any specific vitamin requirements or inherited enzyme deficiencies in the pathway? Organizing intermediary metabolic pathway using these 5 items will highlight the cellular function of each pathway, including its compartment, its energy requirements, its regulation, and any important clinical features relevant to the pathway. Glycolysis is a catabolic pathway that breaks down glucose to produce pyruvate or lactate and produce energy along the way. Glycolysis occurs in all cells, and all the enzymes needed for the pathway are present in the cytoplasm. The net energy output from aerobic glycolysis is 2 ATPs and 2 NADHs. Anaerobic glycolysis produces lactate and 2 ATPs only. And that really brings us to the item number 4, where is going to be very important. These here are rate-limiting enzymes. Step 1, there are 2 of them actually, hexokinase/glucokinase, 2 isozymes, PFK1 and pyruvate kinase. 3 regulated steps. Those steps you want to really pay attention to. Okay? So this is a quick summary here of some of the important, these are the 3 enzymes. Also, you want to add here glucokinase. Don't forget, you have hexokinase, glucokinase. However, only hexokinase is inhibited by glucose 6-phosphate. But remember that in the liver, you're going to have glucokinase. It's not inhibited by glucose 6-phosphate. PFK1 activated by AMP and this molecule, fructose 2,6-bisphosphate, inhibited by ATP and citrate. Pyruvate kinase activated by the product of PFK1, fructose 1,6-bisphosphate, inhibited by alanine, ATP, and pKa-dependent phosphorylation, which is dependent on glucagon, activation of the glucagon receptor. Okay? I want to say one more important thing here. Pyruvate kinase, there's this enzyme, there's a known inherited deficiency in this enzyme. So the effect of this deficiency is to deprive red blood cells of their normal energy complement or needs from glycolysis. And because of that, you get a condition called chronic hemolytic anemia.